Welcome to the Biology Paper 2 revision sessions. I'm going to split Paper 2 down into 8 mini videos that are just slightly shorter and hopefully more manageable. So each topic is going to be 2 videos that are about 5 minutes long. So for the first part then of the first topic, we're looking at homeostasis and response. So just to start off in general then, thinking about homeostasis as a kind of generalised concept. What homeostasis is referring to is the regulation of internal conditions of a cell or an organism to maintain optimum conditions to enable proper function. So it's kind of just about ensuring that at all times body systems are working to constantly maintain kind of the optimum blood glucose levels, body temperature, maintain that at 37 degrees, and just to maintain kind of the correct water and ion levels throughout the body. So whenever those things get too high or too low, the body will undergo a series of processes to basically rectify it back to the normal optimum conditions for function. So when it comes to control systems then, we can split it down into 2 things. We can look at nerves and the nervous system, and we can look at hormones and the endocrine system, because those are the 2 main systems in the body that actually kind of undergo or keep homeostasis functioning for us. So first of all, we're going to look at the basic sequence of events for both parts of homeostasis, and then we're going to focus in on the nervous system. So the basic sequence of events is that initially there'll be a stimuli. So this is something that will change. Okay, so something's happened, something's changed. Receptors will detect that there's been a change. So for instance, it could be that the body temperature has got too high. Receptors on the skin, will detect that, and receptors inside the body. They will then send information to the coordination center, which is usually either the brain, or it can be the spinal cord as well, the CNS. They send information to effectors, which then will do something to bring about a response. So in the case of the kind of raised body temperature, it would be to start sweating to help bring that body temperature back down to what it should be. And that would be the response, would be returning it to normal. So thinking then just about the human nervous system to begin with. So with this one, there's 2 things to bear in mind. Our responses that the nervous system is in charge of are rapid. They're not conscious, so they happen typically, before we've even realised what's going on. And we need to think about a basic reflex arc and to be able to understand this. So for this example, we'll think about kind of what happens when you touch a hot plate. So initially, like we said, in the basic sequence, you've got stimuli, which in this case is the hot plate. Receptors, which would be in this case receptors in the skin on your hand, because obviously that's what you've touched it with. They are going to detect a change in the environment, which in this case is they're going to detect a temperature change, so they'll detect heat. They're going to send a signal via the sensory neuron, which is then going to synapse with a relay neuron, either in the spinal cord or in the CNS, or the brain. Synapsing with that, they're going to send a signal down the motor neuron, which is then going to synapse with some effector cells, which would be muscles in the arm in this case. And those effector cells are going to bring about the response. So in this case, the muscles in the arm are going to either contract to move the hand away from the hot plate. If you're already holding the hot plate, your hand will relax to drop it if it's really that hot. So it depends on the severity of the stimuli as to what your response would be, but you're going to have a really quick reaction to get that plate away from your hand. So the response then is the end result. So it's either that, you're going to move your arm and your hand away from the hot plate, or you drop it if it was already in your hand and then it was hot at that point. So that's your basic reflex arc response. So stimuli, receptor, sensory neuron, relay neuron, motor neuron, effector, and response. Now, when signals are travelling along neurons, they are in the form of electrical impulses. Okay, so electrical impulses travelling along neurons, and when we get to the gaps between neurons, which are called the synapses, what actually happens there is a chemical will be released from the the first neuron, or the neuron that is the signal was passed along. So say the sensory neuron, chemical will be released from the end of the sensory neuron. It will diffuse across that gap, that synapse, and then that will trigger an electrical signal or electrical impulse in the next neuron in the chain, so in this case the relay neuron. Okay, so that's really important though. Electrical impulses along neurons, chemicals diffuse across synapses, which are the gaps. Okay, the next bit then for the nervous system side of things is the required practical that kind of falls into this topic, which is the ruler drop. Now, there's a couple of different ways of doing this required practical. You can have an independent variable. That could be either doing it where you have some people have previously drank something containing caffeine and some people have not, and you could look at the effect of caffeine on reaction time. Or we could do it kind of like we did in class, where we did it based on the amount of practice. So how many goes have you had at trying to catch the ruler? Did you improve? Dependent variable wise, typically what we're actually measuring is the distance the ruler drops or travels, and then we can convert that into a reaction time to see how quickly the ruler was caught. Control variables would be things such as the age, the sex, the mass of the subjects that are participating. There are also kind of procedural control variables, which would be things like making sure, you use the same ruler, the person that's dropping it doesn't give any clues, you use your non-dominant hand, you make sure that your first finger and your thumb are the same distance apart each time, those kinds of things that are just kind of procedure-based control variables. So with this one, it is really important to remember, though, that it is difficult to control a lot of the variables. So, for instance, the ones to do with the person actually participating. Because of that, to make this type of investigation valid, what they'd have to do is do a really large sample size, so have lots and lots of participants, and then calculate averages to actually see, if there is kind of an effect, or if practice has an effect, or if kind of caffeine actually has an effect on reaction time. Okay,